Hello and welcome to Biology class. I am Shipra from ADARSH SHANKAR Mount Abu. For class 11, continue our topic Biomolecules. Today we will study about the enzymes. So first, what are enzymes? Enzymes are the biocatalysts and they are mainly made up of proteins. Sometimes a cofactor is also joined with the proteinase part, which is also known as ecoenzyme. And some proteins are present in the nucleic acids. Nucleic acids are the acids which are present inside the nucleus. So these nucleic acids behave like the enzymes, are known as riboenzymes. So riboenzymes are present inside the nucleus and they catalyze the reactions. And enzymes are the biocatalysts, so it can increase the product per unit time. Like in our body, carbon dioxide reacts with water and it forms carbonic acid H2CO3. But you can see when the enzyme is absent in this reaction, only 200 molecules per hour, is synthesized. But in the presence of carbonic anhydrase enzyme, 6 flat molecules per second synthesized with the help of this enzyme. So, what is the mechanism of enzyme? First, enzyme react with the substrate. React means it is bind with the help of chemical bonds and it forms enzyme substrate complex. Then, in the enzyme substrate complex, the substrate react with each other and convert into the products, and they form enzyme product complex. Okay. Then after some time, these enzymes are separate from the product, and these enzymes are the unused, and it can use again in the different reaction. Now this is the graph between the potential energy and the progress of reaction. You can see this is the first and this is second. In this second, you can see the activation energy without the presence of enzyme is very much, it is large in quantity and then it will convert into the product. But in the, you can see in the first graph, the activation energy with the help of enzyme, it is very low in comparison to the without energy. So, this is the without enzyme. The reaction need high amount of energy, but with the help of enzyme, reaction needs less amount of energy. And with the less amount of energy and large amount of energy, the product is similar. Okay. Now, what is the action of temperature and pH? What is the relation of temperature and pH for the enzyme? Actually, each enzyme shows its highest activity at a particular temperature. Which temperature is known as optimum temperature and which pH is known as the optimum pH. Okay. Now, what is the competitive inhibitor? Actually, competitive inhibitor is the chemicals which are similar like substrate and inhibitor structure is same, but these inhibitor inhibits the enzymatic activity. Okay, so these are known as competitive inhibitor. Now we come to the classification and nomenclature of the enzymes. So first is oxidoreductase. Oxidoreductase is the enzyme which catalyze the redox reactions. What are redox reactions? Redox reaction are those reactions. In these reactions, one substrate can be reduced and the other substrate is oxidized. Like in this reaction, copper oxide is converted into copper. So oxygen is removed. So it is reduced, and in this reaction also hydrogen is converted into water. So oxygen is consumed, oxygen is added, so hydrogen is oxidized. So in the same reaction, one substrate is reduced and one is oxidized. So this is the redox which are catalyzed by the oxidoreductase enzyme. Second is dehydrogenase. Dehydrogenase means removal of hydrogen. Those reactions are known as the reduction reaction, and in these reactions the enzymes work dehydrogenase. Third is transferase. Transferase, it catalyzes a transfer of group. Like when iron is put into the copper sulfate solution, then what is the result? Copper is removed from the sulfate and it forms ferrous sulfate with copper. Clear? This is the transfer of group. Now hydrolysis. Hydrolysis means hydrolysis of ester, peptide, carbon-carbon bond and carbon halide bonds. Then the next one is lyase. Lysis, in this the double bond are formed, like ethyl alcohol. When the ethyl alcohol react in the presence of hot concentrate H2SO4. Hot concentrate H2SO4 is the most, it is the enzyme which catalyzes the reaction, and after that what happens, the water is removed from it and the ethene is formed. Then the next enzyme is isomerase. Isomers are similar in molecular formula, but their structure is changed. Their functional group may be changed. So these are known as the isomers. And like in the glycolysis. Glycolysis is the first step of respiration. During this space, DHAP, dihydroxyacetone phosphate, is converted into PGL, and PGL also can convert into the DHAP. So both are the isomers and they can interchange. So isomerase enzyme is catalyze this reaction. And the last one is ligase. Ligase, during the DNA replication or duplication, one strand of DNA is continuously form the other DNA, but the second one is formed in the Okazaki segments. These are known as Okazaki segments. Okay, the second strand is formed in the Okazaki segments, and these segments are connected by the DNA ligase. So the function of DNA ligase is joins the Okazaki segment, joins the DNA segment. So this is all about the enzymes.